It's within the LDOT HOD liquid biobanking, there's plasma.
Okay. Good. Okay, thank you very much. Anil, did you want to, are you still, I don't know if you're still on.
Yeah.
Yeah, great.
So thanks, Sanina. That was wonderful. Next, we have Dr. Liza Pon. She's a professor of pathology and cell biology. Her own research is in mitochondrial biology, looking at organelle quality control and interaction of mitochondria with the cytoskeleton and other organelles. And she's extensively funded through the NIH, including an R35. As a member of our cancer genomics and epigenomics program, Liza is also director of our Confocal and Specialized Microscopy, or CSM, shared resource. Liza, I'll turn it over to you, and thank you as well.
Okay, great.
Lisa, we're actually seeing the presenter mode on screen. At least
you
that's what
are?
I'm
Okay,
seeing.
so,
Yeah, so you might need to go to, there
better?
we go. Yeah,
Okay.
perfect. Thank you.
Okay, so thank you for the introduction. Oh, can everybody hear me and stuff? Emer can, anyway, all right. I'm assuming.
Yeah, we can hear you.
We can hear you.
All right, so I'm here today in my capacity as the director of the Confocal and Specialized Microscopy resource, a shared resource. And you may not know me, but I'm sure every single user knows Teresa Swain and Laura Mutznyanyu. These are, Teresa is a cell biologist, Laura is a biophysicist. They have decades of imaging experience and serve to manage and co-manage the confocal facility and also as imaging specialists. So the overall mission of the Confocal Shared Resource is basically to provide our users with the technology and the scientific expertise to see things that no one else has seen before. And we're equipped to do so. So we have wide field imaging, both conventional and higher resolution digital deconvolution. Our microirradiation and laser dissection system is a multi-purpose instrument. In the microirradiation mode, the laser can be used to irradiate a sample for applications including inducing DNA damage. And then the microscope can be used to visualize the cellular response to that insult. In the laser dissection mode, the laser can be used to excise a region of interest from heterogeneous tissue, whether it's a stem cell niche or a tumor. And then the laser is used to catapult the excised sample in a contact-free, sterile manner to a receptacle for further analysis by genomics or proteomics. We have a ton of confocal microscopes. We've also recently acquired super resolution imaging systems, including structured illumination imaging and stochastic optical reconstruction microscopy. Long names, but the long and short of it is that the super resolution imaging systems allows us to have 2- to 3-fold increased resolution compared to that obtained using confocal or conventional wide field imaging. And today, what I'd like to talk about are some of the instruments that we're using for live cell imaging. Now, what you're looking at, or we'll see, is like a classic live cell imaging movie, which shows you a macrophage moving towards and taking up pathogens and then degrading those pathogens in the lysosome. So to orient you, the pathogens are shown in green fluorescence. Here you can see the macrophage, and the red fluorescence is actin within the macrophage. And what I trust you can see is the actin at the leading edge of the cell as it moves towards the pathogens. You can see these phagocytic cups that are coated with actin at sites of pathogen uptake. And then you can see the actin comet tails. I think you'll see one here, which actually propel the pathogens from the cell cortex to the interior where they're degraded by the lysosome. So basically all live cell imaging studies require a couple of things, and that is basically to maintain cell viability and cellular function. And this requires environmental and temperature control. It also requires using illumination and imaging conditions that provide the spatial and temporal resolution needed to detect the activity of interest without damaging either the cells or the fluorophores within cells. So what I'll be talking about today are instruments that we have for high resolution, high speed live cell imaging, and then for long-term live cell imaging. So as you probably know, the optimal sample for optical imaging is 2-dimensional. This is why people like to visualize fibroblasts or squamous epithelial cells. The cells are flat and effectively in one optical plane. However, as you may know, biological samples, whether it's a cell dividing or a tumor or an organ, are three-dimensional. And so using wide field microscopy, you basically illuminate the sample and you get the image in your focal plane, but you also get out of focus fluorescence from above and below the sample, which compromises image resolution. So in about 1955, Marvin Minsky, then well known for his studies in artificial intelligence, built the first confocal microscope. And in this, and using laser scanning confocal microscopy, the incident light, that is laser, was focused into a very, very tight beam. And then a small pinhole was inserted before the detecting site, which allows you to filter out all out of focus fluorescence, which basically allows you to get resolution like this with laser scanning confocal microscopy, as opposed to this, which you can see in wide field. So this is why people use confocal microscopy a lot, but in terms of live cell imaging, it has a number of limitations. And that is, for laser scanning confocal microscopy, you have a point source that basically scans across the sample, point by point. And that works, and it gives you really high resolution, but it's very, very slow. And in some cases, slower than what you need to capture a living, a live cell event. In addition, the laser is very, very intense, and it can be damaging. So a lot of these problems are overcome using the spinning disk confocal microscope. So it is a confocal, that is, you have a single laser source and you have a pinhole in front of the detector, but instead of just having one laser source and one detector pinhole, you have many, many, many sources and detectors that are inserted into a disk that spins. So what you get then is the resolution, or close to the resolution, of laser scanning confocal microscopy, but you get speed. In addition, with spinning disk microscopy, there are micro lens arrays that are very effective in capturing the emitted light. And as a result, the incident light is much lower, and that results in less cellular damage and preservation of signal. Okay, in addition, of course, for live cell imaging, you have to have environmental temperature control. And so here you can see the stage top chamber that we have that controls both temperature and the atmospheric conditions. Okay, so why do you need live cell imaging to be so fast? So one example is translation. So as you may know, or you may not, during protein synthesis, amino acids are added to a nascent polypeptide chain at a rate of 2.6 to 5.6 amino acids per second, okay? So you need to be able to do very rapid imaging. In addition, there aren't all that many translation events that are happening within cells, so you need to have an ability to amplify the signals at translation sites and to detect them. And what you can see here is the approach that Ben Hobson in Peter Sims' lab used, the SunTag, as a translation reporter system. So what they did for these studies was basically to take multiple copies of an epitope, in this case peptides from the GNC protein of yeast, and insert it into a coding region of interest. They then simultaneously expressed an antibody which recognizes the epitope and is labeled with GFP. So that allows you to detect the protein. Now, to ensure that you're looking at translation as opposed to full-length protein, what they did was they inserted an auxin-inducible degran, that is an inducible degradation system, at the C-terminus of the protein. And as a result of that, what happens is that all full-length protein is degraded, and the only thing that's left behind are nascent polypeptide chains. And so what they did was they expressed this system in cell culture, and then they used the spinning disk confocal to do rapid, high-resolution live cell imaging. So to orient you, what you see on the left is the cell. Here is the nucleus, here's the cytoplasm, and all these punctate structures that you can see are sites of translation. And so they imaged this cell actually at 2.5 to 10 second intervals for a total of 10 minutes, or 8 minutes actually. And what you can see is multiple putative translation sites within the cell. To ensure that these were indeed translation sites, Ben treated the cells with puromycin, which is a translational inhibitor. Here you see that cell prior to puromycin treatment. You can still see translation sites in the cytoplasm of the cell. But with time, you can see very, very rapid inhibition of translation by puromycin and loss of the signal. Okay, so for imaging, it's not just about getting the images, it's about quantitative analysis. And so what Ben did together with Teresa and Laura in the Confocal facility is to use public domain software, ImageJ, provided by the NIH, to automatically identify punctate fluorescent structures in the cytoplasm, and then to count those structures as a function of live cell imaging. And here you see the results of that quantitative analysis. The red tracing here shows you the control. And what's important is that the number of punctate structures persist throughout the imaging time, which indicates that the imaging conditions didn't compromise translation, which is of course very important. Equally important, you can see that treatment with 3 different translational inhibitors blocked the translation, that is, reduced the number of punctate translation sites, and did so with different kinetics. So this is an example of rapid high-resolution live cell imaging. We also have a newly acquired system for long-term live cell imaging, which is the Citation 5 system, which we obtained using funds generously provided by the Cancer Center. So here you see a photograph of the system, and it has a number of components. This is the imaging platform, which is where the samples are imaged. It's temperature controlled. It also has LED illumination for visualization of fluorescence, blue, red, green, and far red. Objectives from 5x to 60x, as well as a rapid sensitive CMOS camera. This here is what they refer to as the BioSpa, which is an incubator that is, of course, temperature and environmentally controlled, that can house up to 8 different samples simultaneously. And then the samples are transferred from the BioSpa to the imaging platform using this robotic arm. So here you can see the system in action. You can see the drawers of the BioSpa opening. We can basically accommodate just about anything. And then you can see the robotic arm transferring the sample to the imaging platform. So I should say that this is maybe a 6-well plate. We can accommodate basically every commercially available plate or flask, or anything that can be generated by 3D printing using the in the Citation system. Okay, and so I want to show you some examples of use of long-term live cell imaging. And the first is organoid formation, which was visualized by Alex Devani in Laura Kaufman's laboratory. As you know, organoids are organ models, which are used to study organ differentiation, physiology, and dysfunction. And what Alex did here was he basically co-cultured cultured breast cancer cells that were differentially labeled, and then visualized organoid formation over time using the Citation system. And as you can see here in day 1, you can see relatively dispersed or distributed cells. The red cells are one breast cell cancer line, the green are another. However, what I trust you can see is that over the course of time, you can see segregation of those cell types and organoid formation, such that you have one population mostly at the organoid periphery and another within. The next slide illustrates the multicolor imaging capability of the Citation system. What you're looking at here is an embryoid body generated by Bo Li from Michael Shen's lab. And in the left, you can see that embryoid body, which is an intermediate in organoid formation, which is generated from mouse epiblast cells. And here you can see the 3-dimensional organization of that embryoid. And in the right, in high magnification, you can see specific cells and components within those cells visualized using staining and immunofluorescence. So the blue shows you the nuclei stained with the DNA binding dye DAPI. They were also able to detect multiple neuron-specific markers, including double cortin, nestin, and neuron-specific tubulin isoform, which were visualized in green, red, and blue, respectively. Okay, moving forward, another example of a type of long-term live cell imaging that can be done using this system is to look at cell migration, which of course is critical for understanding metastasis. So in this case, cells were cultured to form a monolayer in a well of a 96-well plate, and then a scratch was applied to that plate which cleared out the cells. And in fact, we have a auto scratcher, which will produce exactly the same scratch in all 96 wells. So you get a system in which you have a cellular areas here and here, and then a cell-free area. And then long-term time-lapse imaging was used to visualize cell migration into the scratch area. And what I trust you can see is that the cells do indeed migrate. They obtain these images at 15 seconds over the course of 12.5 hours using high contrast imaging. Okay, that's it again. Okay, and of course, it's not just acquiring the images, it's quantitative analysis. So we have software in the system to do cell counts, to quantitate cell counts and morphology, to quantitate fluorescence intensity and co-localization, and to track particle movement and cellular movement. And on the right here, you can see some examples of that. So basically, using our software, you can define cell boundaries, as you can see here. You can define intracellular structures, including the nucleus. In cells where you define the cell boundary and the nucleus, you can, of course, also define the cytoplasm. And you can also identify structures of various specific sizes and fluorescence within the cell. And so you can quantitate cell morphology, cell size, cell number, and you can also quantitate the fluorescence and co-localization of fluorescence within cells. And so here you can see quantitation of the analysis of cell migration in movies similar to the one I showed you previously. So what you're looking at is analysis of migration of either untreated cells or cells that are treated with different concentrations of cytokeratin D, which inhibits cell migration by perturbing the actin cytoskeleton. You can see that we can get good quantitation of cell migration, as well as a dose-dependent decrease in that process as a result of cytokeratin D treatment. Okay, and finally, our users obtain data in the confocal facility, not just because we have the equipment, which is spectacular, but because we have the scientific and technical expertise to help our users make their experiments work. So here you can see us, this is me, ignore me. What you should focus on are Teresa and Laura. What Teresa and Laura do is, of course, they maintain the equipment and provide access to it, but they also provide guidance to our users at all sorts of different levels. So for experienced users, they can train them on instrument use and write scripts for quantitative analysis of the images obtained. We also welcome newcomers of all sorts. And for inexperienced users, Laura and Teresa will basically provide every bit of information you need. What fluorophore to use, what cover slip to use, what microscope to use. They will provide information on protocols or get you in contact with people who are carrying out similar experiments so that they can provide guidance on protocols. They'll even sit at the microscope with users to obtain images, to save them, and then to work with users for image enhancement and analysis. So what I want to encourage everybody here to do is to carry out either live cell imaging experiments or other imaging experiments in the confocal shared resource, in part because we have really wonderful state-of-the-art equipment, but in part because we have the scientific and technical expertise to make your experiments work. And so to find out more about the confocal facility, please refer to your website, and I guess I'll take any questions. Thank you.
Thanks so much, Liza. That was terrific. You also get some of the prettiest pictures for our websites and posters and everything else. Okay, if anyone has any questions, please pop them into the chat or raise a hand. We have a lot of participants on the call, so we welcome any questions for Liza and the confocal shared resource. Okay, we'll take a couple of minutes, see if anything's coming in. Great. One question, great presentation. We, the question asker, Lance Cam, are located on the downtown campus. Is there a cell culture facility that can hold cells before imaging?
Yes, in fact, we've moved to our new site in the Lasker building. It's on the 3rd floor of the Lasker building. And we do indeed have a cell culture area that people can use for their live cell samples. Generally what people is they set up the samples, they move them, put them in the cell culture area let the cells settle down, and then use them for imaging experiments. So yes, that's readily available for all users.
Super, thank you so much. I don't see any other, oh, how about, so somebody's asking me a question about 2-photon imaging. If you could mention about 2-photon and your capabilities.
Yeah, so we do indeed have a multi-photon laser scanning confocal microscope. It actually was just upgraded recently so that we have new lasers, new detectors, super fast resonance scanner, and it's readily available for conventional 2-photon imaging so yeah, please feel free to contact Teresa and Laura about that microscope and how to use it and how to sign up for it when you want.
Great, terrific. Perhaps, Liza, you could mention a little bit about, and you probably might have mentioned this and I missed it, but about your current availability during this time of ramp down and how folks can use the resource.
Yeah, so basically, we're pretty much shut down in the confocal facility, and Teresa and Laura are working remotely. They are working extensively with our users in image analysis and quantitative analysis. We do make our microscopes available to individuals who are doing COVID-19 research, and there are indeed some people who do it. So some microscopes are being used, but in general, our microscopes will not be accessible for users who are not doing COVID-19 research until we ramp up. On the other hand, Teresa and Laura are always available to talk to you about experiments that are either ongoing, under analysis, or in the planning. And you can reach out to them at their email, probably from 9 to 5.
And those are on the website, on the landing page on the website for the shared resource. Okay, is there any other questions from the group? Oh wait, let's just see. We have another question. Oh yeah. I'm sorry, we have a couple more questions. Can you image uptake of macromolecular complexes, FITC conjugated in real time?
We're equipped with turf microscopy so that we can see uptake of things at the cell surface. So yes, and we also I'm not sure how quick the uptake is, but yeah, the turf is probably the best application, and we're certainly equipped to do that.
Great. Follow up on the first question, what's the maximum amount of time that cell culture can be used prior to imaging?
Right now, I would say that we should have a sign-up sheet for use of the cell culture space, but as far as I know, it's not super heavily used. So people should feel free to sign up for use. In general, we don't like any person dominating any one piece of equipment, or service provided, but right, as long as the time is available, people can use it.
Great. Live cell in vivo imaging, it's a difficult consideration with in vivo animals. Do you work with that, or is it something outside the shared resources purview?
We don't have a system for in vivo imaging in live animals. However, Joji Furasaki, I guess his last name is, does have a core that he is running right now.
Great, and excuse me, I have one other question here. What's the maximum amount of time the citation 5 can be reserved?
Okay, so, this was, we purchased it in part, we obtained it in part because it is indeed a true multi-user instrument. And so basically up to 8 different users can use it at any point in time. The system is set up so that we can coordinate the time-lapse imaging so that we can fit everybody in and they can obtain the images that they need. So there is no absolute maximum amount of time. That said, we want to encourage people to do long-term time-lapse imaging. So in contrast to every other microscope that we have, where we charge for the amount of time that you're on the instrument, for the Citation 5 instrument, we don't charge for the amount of time that your samples are actually in the incubator. We only charge for actual microscope use time. So that again is to encourage people to do long-term live cell imaging.
Wonderful. Well, thank you very much. I really appreciate our 2 speakers being here today and telling us about their shared resources. I'd like to remind everybody that we are running these resource Resources for research every Tuesday, 4 to 5. Next week, we'll hear from 2 more of the Cancer Center shared resources, the biomedical informatics and cancer biostatistics. We also hold seminars every Wednesday from 4 to 5 via Zoom. We have one tomorrow, which is from our office for research at the medical school. And that will be from the Director of Research and Strategic Initiatives with some great information on grants, resources, education, and tools. For today, please everybody watch for a survey coming to you from Stuart Fisher. We like to get your feedback, understand how we're doing and how we can do better. And once again, thank you so much to our speakers, a really enjoyable session, and thank you to everybody for joining us. Okay, take care everyone, stay safe.
Thank you.